Okay, so we are starting our genetics unit, and this is the first video that you guys are going to have to watch. And it's the most simple form of genetics, and we're going to do a little bit of the history of genetics before we get started doing those practice problems. So there's going to be some words that you need to write down as we go through, and you should have your vocab sheets out with you because it's going to be important since you're not actually taking real notes on this PowerPoint. So by the end of this, you're going to be able to give us a little bit of history about Gregor Mendel, who we're going to talk about in just a second. You're definitely going to have definitions for these words: genes, alleles, genotype, phenotype, among some others. And hopefully be able to do, after you practice a little bit, some one-trait crosses using Punnett squares. And hopefully that's going to ring some pretty big bells because you did that in like 7th grade. So it should be something that you already know how to do, you just might not remember yet that you know how to do it. All right, so the history of genetics. In the 1800s, 1822 to 1884, this guy named Gregor Mendel, who was an Austrian monk, and his little picture is right here below, obviously not totally accurate, but he was an Austrian monk who, among other things, ended up studying pea plants. So he was also in charge of, gardening, and peas were something that the monks ate. And if you've never seen a pea plant, they have lots of different traits that you can look at, like the color of the flowers, how tall the plants are. They can either be tall or short. The peas themselves can be different colors, yellow or green. They can be round, like the ones we see in the grocery store, or they can actually be wrinkled, so they kind of look very shriveled up. But he studied them, and he looked at the traits, and he's actually what we call the father of genetics. He figured basically out how inheritance worked by using this very, very simple system. And there's an actual portrait of Gregor, so a little bit looks similar to the one in the corner here. And if you want to watch this video, My Favorite Scientist with Gregor Mendel, we will also link that up for you. It's actually a cute little video. So let's talk about some very basic vocabulary. So what is a gene? You've definitely heard this word before. A gene is a sequence of nucleotides. So remember those A's, T's, G's, and C's in our DNA on a chromosome that codes for a specific protein. So one gene is going to give the information to make one protein inside of the cell. And an example that's really easy is the eye color gene. So we're all familiar with that. We know that you have eye color, and there's a gene inside of your cells that codes for what color your eyes are going to be. But you should be thinking that, well, not everybody has the same eye color. So we have like blue eyes and brown eyes and hazel eyes and green eyes or gray eyes. So there must be something going on there. And you'd be right. So everybody has an eye color gene, but they might have a different version of that gene, and we call that an allele. So take a minute and make sure you write both of those definitions down, the one for gene and the one for allele, and make sure you write your examples. So if you want to use eye color gene for your example for your gene one, that's fine. And then for alleles, here you go. So you could have a blue allele. You could have a brown allele, a green allele. And you get these alleles from your parents. So your parents are giving you the genetic information to make these things. That being said, you have two parents. So for every single one of your genes, you inherited 2 alleles. So mom gave you a gene for eye color, and dad gave you a gene for eye color. And how that happened was the gametes, remember from meiosis, the egg and the sperm, fused together to make a zygote. And dad's 23 chromosomes that he gave you and mom's 23 chromosomes that she gave you came together, and that combined, only when that combined to the full 46 chromosomes that we find in humans, we had the entire recipe to make you who you are. And remember, those are called homologous chromosomes. So I'm going to show you a picture up here. So if you look, this is a very basic picture, But let's say you have this gene from mom and this gene from dad. We know they're homologous. Homo means same, log meaning information. So they're going to carry the same information. They have a gene for eye color, a gene to make a specific enzyme, a gene for something called cytochrome C, which is used in cellular respiration. So again, you get eye color, one from mom and one from dad. And depending on which ones you get from your parents determines then what color your eyes will actually be. And if you look, here's a beautiful picture of the range of eye colors that we can see for humans. And I'm sure there are some there that are not covered. But just, you know, the different alleles that we inherit give us this beautiful range. So let's kind of circle back to Mendel now that we have an example of what genes are and what alleles are. So we have this picture right here. So there are two flowers. So pea plants have two options or two alleles for flower color. So you can either have purple flowers or you can have white flowers. And whenever Mendel, because he was working with these pea plants, whenever he mated in his first generation, which we call the pea generation or parent generation, whenever he mated a purple plant and a white plant, all of their offspring were purple. Every single one of their offspring were purple, no matter how many times he did it. And he did it a lot. He ran like 20,000 experiments over his lifetime. So he did this a ton. So he had a lot of data. And then all these purple offspring, if he let them self-fertilize, because sometimes plants can do that, but it's still two gametes coming together, the results, and again, these are the offspring of these two plants up here, were 75% purple flowers and 25% white flowers. So if you look, here's just a visual representation. You have your purple and your white parent flowers. All of their offspring in this F1 generation, which we call that first generation after the parents, they're all purple. Again, no matter how many times he did it, they were always purple. And then when he let them self-fertilize, again, so, you know, there's probably hundreds of these flowers around. When they're mating with each other, he got this kind of breakout, the 75% purple to 25% white. And again, no matter how many times he did it, it would always come out to be about 75-25. So he got to thinking about, what does this actually mean? Because he knew something was happening and he just had to put the pieces together. So this is what he learned. Some traits, like flower color, purple, some traits are dominant over others. So he said purple was dominant over white. And the white trait we call recessive because you have the dominant trait and you have the recessive trait. Alleles, remember your versions of genes, can be dominant. And that means if you inherit one copy of that dominant allele, either from mom or from dad, you're going to show that trait. So let's say the trait is being tall. If you inherit one copy, either mom or dad gives you the copy of the tall allele, you're going to be tall. On the other hand, the recessive trait is only ever shown if you inherit two copies of that recessive allele. And that means mom had to give you a copy and dad had to give you a copy. Because, again, like we said, if you inherit one of these, one copy of the dominant, you're going to be or show the dominant trait. So you need to actually inherit two copies of the recessive allele for it to be shown. And in this example, it would be short. So you'd have to inherit a short allele from mom and a short allele from dad to be short. Okay, another vocab word. You have a genotype. So you have a genotype for all of your genes. And that's what combination of alleles were inherited for a gene. So you have a genotype, again, for all your traits. So remember, you get two alleles for all of your traits. Let's say we're talking about heights, and we're simplifying this down. So height is the trait that we're looking at. If you inherit two copies of the dominant allele So mom gives you a dominant and dad gives you a dominant. We call that genotype homozygous dominant. So the prefix homo means same. So your genotype would be big T, because we use the capital letter of the trait for the dominant allele. It's kind of a shorthand. If you are heterozygous, that means you have inherited one copy of the dominant allele and one copy of the recessive allele. So one parent gave you the dominant allele, one parent gave you the recessive allele for the particular trait. And it would look like this. So your genotype would be big T, little t. And then you could also be homozygous recessive. And you inherit two copies of the recessive allele. Again, homo meaning same. So your genotype would be little t, little t. So before you move on, make sure that you take a moment to write down the definition of genotype and your examples. You also should be writing down the definitions of homozygous dominant, heterozygous, and homozygous recessive. So you might want to pause for a second so that you can write those all down before you move on. All right, so if you're looking at this heading, you say it says genotype and phenotype. We talked about genotype, which is the combination of alleles you inherit from your parents for a particular gene. But phenotype is the physical manifestation of a genotype. So let's work through some of examples. So this is going to be exactly the same as the slide before. Okay, so if you inherit two copies of the dominant allele, so you're homozygous dominant for your genotype is big T, big T. But what does that actually mean? So remember, what this is actually saying is you've inherited two copies of an allele for being tall. So your phenotype would be tall because you have at least one copy of that dominant allele. If you inherit one copy of the dominant and one copy of the recessive, that means, again, vocab word, you're heterozygous for your genotype. So big T, little t. One parent gave you the dominant, one gave you the recessive. But because you still have a copy of that dominant allele, you're still going to be tall. You only need one copy of it. So it doesn't matter if you have one copy or two copies. Because it's dominant, you're going to be tall. If you inherit two copies of the recessive allele, so your genotype would be little t, that's the only way in this example that you would be short. So both parents would have to give you that gene. So again, there's some vocab words on here. Make sure you write down your definition, of phenotype and your examples. Okay, all right, so tall plants, possible genotypes. So take a minute, think about it. What are your possible genotypes if you are a tall plant? And hopefully you are thinking big T, or big T, little t. Because again, in this situation, you only need one copy of that dominant allele to show that phenotype. And the phenotype then, physical manifestation of the genotype, which is right here. So here's our genotype. What does that mean? It means that the plant is going to be tall. Short plants, possible genotypes. Hopefully we got that one right. It's little t. And then the phenotype for that, again, physical manifestation of the trait, the plant is going to be short. So it's just kind of running back through genotype and phenotype. Okay, so just to kind of sum all this up so far, we have an egg, which is haploid. Remember, that means it has half of the genetic material for an organism. And the sperm over here is haploid with the other half of the genetic material for an organism. So when they come together, they make the zygote, which will turn into the offspring, and we have two copies of each chromosome. So this is just review. Individuals are diploid. You are diploid in every single one of your cells except for your eggs and your sperm. So you have two alleles for each gene because you have two parents. The gametes then, as it is shown in the diagram, are haploid because each parent needs to contribute half of the genetic material. So each gamete has one allele for each gene, and then when they come together, you have two alleles, again, for each gene. And because we can't control what genes are passed on, It's going to be totally random which allele for each gene gets into that egg and into that sperm, and eventually which egg, and sperm actually come together. Which is why if you have siblings and you have the same parents, you probably don't look identical, because it's different every single time that egg and sperm are made. So it's actually the advantage of sexual reproduction. So now we're going to practice some crosses. Again, this should be exactly like what you remember from 7th grade. The word up here might be new to you, monohybrid. So mono meaning one, hybrid meaning we're crossing two things. So we're looking at, it's just a fancy way of saying we're looking at one trait at a time. So mono, one trait. Okay, so like we just said, gametes can only have one allele for each gene. So let's say we have a parent that, because remember, they have two parents, so their genotype is big T, big T. And let's say it's tall, right? So this parent's going to be tall, but they're homozygous dominant for that gene. So the gametes they can make can only be, homozygous recessive, so I'll put two parents up. The gametes for all of these, if I am homozygous dominant, my gametes can only ever have that one trait, because that's literally all that organism has. They only have one version of that gene because they're both the same. So this and this version are exactly the same for the gene. And if you're smart, you're thinking, oh, homozygous recessive parent, that's going to be the same thing except with little t's. Because again, any egg or any sperm that they make is only going to get that copy because that's all they have to give. But here is where it gets interesting. This heterozygote parent. They actually have two different versions in their cells. They have two different versions of this gene. So when they go to make their gametes, half of them are going to be big T and half of them are going to be little t. So we're going to practice. So kind of ignore that visual at the bottom for now. So when we do a cross, we're determining all the possible combinations of gametes. So I know the two letters at the top, big T, and big T, little t, do not match up with this diagram down at the bottom. But what it's showing you is, if you look, that let's say this is mom's. Mom is heterozygous. So half for this particular trait. So half of her eggs are going to contain the dominant trait, and the other half are going to contain the recessive trait. And we can tell that by the letters. The sperm over here for dad, he only has the recessive trait. So he was homozygous recessive for this particular trait. And what the Punnett square is actually showing us is that if this sperm meets this egg, then this will be the genotype of that offspring. If this sperm meets this egg, this will be the genotype of the offspring. This egg, this sperm, this will be the genotype. This sperm, this egg, this will be the genotype. So we're kind of seeing all the possible outcomes of the combinations. So these are the offspring in here. These are the possible offspring. Every single time these two parents would have a baby, these would be the chances of the genotypes for the offspring for that one trait only. Okay, so I kind of just explained all this. You can ignore what's over there. Okay, so we're going to practice one. So if you want to take a moment to you don't have to do it on your vocab sheet, but to draw a square out, that would be great. So we're going to cross two parents. One is homozygous dominant for a trait. So let's say it's a plant and we have tall versus short. So we have one parent that's tall, big T, and one parent that's short, little t. So my big T parent, it doesn't matter which gender it is, its gametes are going to be, no matter what, because they're homozygous, they're going to all be the same. So it's going to be big T or big T. And we're going to put them right there. So one kind of over each column. The other parent is going to be the exact opposite. So little t, because again, all the gametes they can make are the same, and they're going to go over here. So again, you put the genotype of the parents along the top and along the sides. It does not matter which parent goes where. And then all you have to do is fill in the boxes. So if you take this big T and put it down here, and this little t and put it over here, this combination would be heterozygous, big T, little t. We take this T and this T all the way down and over. Same here, big T from here goes here, little t goes here, and then last one, this one here, this one here. So all of the offspring, their genotype would be big T, little t, 100% big T, little t for genotype. And then their phenotype, the physical manifestation of the trait, would be 100% tall. So all of these offspring would be tall, even though they had one tall parent and one short parent. So now let's do another cross, big T, little t with big T, little t, so two heterozygous parents. So again, tall and short. All right, gametes from parent 1, gametes from parent 2. So parent 1, it doesn't matter which one's which because they're both the same, they're both heterozygous. So let's say it's this parent. Half of their gametes would be big T and half would be little t. And same with parent 2 because again, they're heterozygous. So this is going to be the square right here. So again, you put your parents along the top and along the sides. And then I'm going to give you a second to pause maybe the video and do this out. Okay, so if you pause, let's check your work. So in this corner, it would be big T, so you have a homozygous dominant offspring. Big T, little t, big T, little t, and then little t. Sorry, I went too fast. So if you were talking about, I'm just going to put all these up. So the genotypes you would see, so this goes for the Punnett square that was just on the screen. All the genotypes we saw for the offspring, they could be big T, that was that top left corner, big T, little t, which went diagonally across, and then little t, the bottom right-hand corner. And then the ratio of the genotypes, you would write out like this, 1 to 2 to 1. So 1 big T, 2 big T, 1 little t. And then the phenotype ratio, remember, we're either going to be tall or short. So the phenotype ratio, we'd actually have 3 or 75% tall offspring and 25% short offspring. So remember, these right here, big T and big T, little t, those are going to be tall because you only need one copy of that gene. This one's the only one that's going to be short.